You know, sometimes in research, all the pieces are right in front of you. You just have to find out how they fit together. Today, on World Cancer Research Day, we are bringing you a very special episode. It's all about multi. Well, that's multianalytes, multiomics, and multimodalities in liquid biopsy analysis. I know that's a lot of M's we're going to talk about today. So stick with us. It's time to get curious. Welcome to Q-rious, the show where molecular mysteries meet modern miracles. Today, I'm joined by my colleague Marcus, who is heading up the Sample Technologies Development Team at Pre-Analytics, a QIAGEN and BD joint venture. Well, welcome, Marcus.
Thank you so much, Kirchi, for having me.
Well, speaking of M's, Marcus, you're going to touch upon three of them today: multianalytes, multimodal, and multiomics. So what exactly are these?
Yeah, maybe let's start with multianalyte. So at multianalyte analysis, we look at different biomarkers or biomarker classes at the same time. So it could be free circulating DNA, free circulating RNA, or circulating tumor cells, for example. We're looking at a set of genes of interest or mutations, or a set of genes being expressed. But it's a panel of predefined biomarkers you're looking at the same time.
Interesting. And what about multiomics then?
Yeah, in multiomics, it's a bit larger, the view. We look at the entire transcriptome or the entire genome, and then multi means we look at transcriptome and genome at the same time. So that's the main difference to multianalyte, where you only look at a panel.
And talking about the third M, then the multimodal, what's with that?
Yeah, the multimodal adds another level of complexity. Here we are looking, for example, into different sample types from the same donor. For example, looking at blood and urine at the same time, and then combine it with multianalyte or multiomics to look at RNA, DNA panels, or whole transcriptome, whole genomic analysis.
So maybe can you explain a little bit why is the multimodal approach gaining so much traction for liquid biopsy analysis these days?
Yeah, sure. You can compare it maybe with looking at a picture in 2D or 3D. If you look at the multianalyte or multimodal or multiomics approach, you see what's going on in the body from a different angle, and that gives you a much more nuanced and complete picture of what's going on in the body.
Right. And you've been long enough in the field, so do you think this is really reshaping the way we think about liquid biopsy overall?
Yeah, absolutely, because I think the deeper insight allows really insights which haven't been possible in the past. It allows, if you combine those different methods, it allows early cancer screening or detection of cancer early on. It allows disease progression, and it allows predicting also how disease is going on. At the same time, it's developing beyond oncology. So prenatal diagnostic was the pioneer, the first application. Then oncology is the second big wave coming now. But we see a lot of research in other fields like organ transplant rejection, or like inflammation, neurodegenerative diseases, or many other application fields.
Talking about the multianalytes earlier, right? So examining these multianalytes must be really complex. There must be really inherent challenges in the analysis. So can you share a bit about that?
Yeah, absolutely. I think the biggest challenge is really that the different sample components have a different stability.
Right.
Let's start with cfDNA, free circulating DNA. This DNA is released from dying cells in the bloodstream. For example, if you have a tumor cell circulating in the bloodstream and it's dying and degrading, the DNA is released. At the same time, the body is eating up those DNA fragments, and also other cells like hematopoietic cells are releasing free circulating DNA. That leads to that the tumor signal is actually getting smaller over storage, and other signals are coming up. For exosomes or extracellular vesicles, they carry RNA, for example, as the most interesting cargo. And here is a similar picture. Cells are releasing new exosomes and vesicles, and by this also changing the RNA transcription profile in the bloodstream, while other exosomes are absorbed by cells and are removed from the fraction of cell-free nucleic acids. And then last but not least, for circulating tumor cells, they are carrying epitopes, and those epitopes are often used for isolation of circulating tumor cells. These epitopes can also change over time of the disease, and it might lead to the fact that the cells are not captured anymore and are not found.
Right. You just reminded me, it sounds a little bit like juggling Fabergé eggs, really. If you let one drop, you waste something really precious.
Yeah, indeed, it's like this. And that's why it's so important to really try to combine all these things in a workflow. We have teamed up to develop standardized workflow to actually do all these analyses without wasting any of these analytes. So we started with two PAX-gene blood cfDNA tubes, and we're first of all isolating the circulating tumor cells with the ADNA Select CTC method. And then we were isolating the RNA and the DNA out of these cells with the all-prep nano kit. From the remaining sample, we were able to generate plasma and then isolate the circulating cell-free DNA by using the Kaya Minilute cfDNA kit. And we were able to isolate the genomic DNA from the Buffy code fraction as a reference for the analysis. And then we took the second tube to isolate the plasma and get the exosomal RNA out of this.
Wow, all of this is happening from a single blood draw. That's amazing, right?
Yeah.
That's progress. Can we use the samples that are drawn from like normal EDTA tubes? Is that also possible?
Yeah, that's possible too. The only downside of this approach is that EDTA plasma is only stable for 2 to 4 hours at 2 to 8 degrees. So you need to do the analysis right away, and you need to cool the sample all the way. While, for example, with the PAX-gene blood cfDNA tube, you can store the sample up to 10 days at room temperature, 15 to 25 degrees.
Okay. And so that's all about stability. And in this workflow, as I understand, so do you have to do all of the steps immediately, or are there really stopping points at any part of the workflow?
Yeah, that's an important question, because it's many workflows you have to do in parallel, but there are really easy stopping points. For example, in the CTC workflow part, you can freeze the lysate as soon as you have selected the CTCs and lysed them. So the DNA RNA isolation can be parked. For both the cfDNA isolation and the exosomal RNA isolation, you can freeze the plasma and then collect samples in the freezer until you have a sufficient batch size, and then do them all at once.
Interesting. So for people who would like to kind of see this more visually, let's take a look.
With the growing number of publications in recent years, a challenge with liquid biopsy is choosing which analyte to isolate between circulating tumor cells, cell-free DNA, and circulating RNA. A single analyte won't tell the entire story, and blood samples are often insufficient to investigate all of them. Make the most out of your sample and complete the picture with our multi-analyte liquid biopsy workflow. How does it work? Start by using only 2 PAX-gene blood cfDNA tubes, or EDTA tubes. CTC nucleic acid samples collected using PAX-gene blood cfDNA tubes are stable for up to 72 hours. You can use the ADNA-test CTC Select to isolate and enrich CTCs from whole blood sample. Afterwards, the same blood sample depleted of CTCs is used to generate plasma and extract cell-free DNA using the Kaya Minilute cfDNA kit. To complete the picture, circulating RNAs are extracted from the second blood tube using the exo-RNase kit. The multi-analyte workflow also offers the option to freeze samples at certain steps, which allows convenient planning of laboratory work. Some steps can also be automated on the Easy-to-Connect or on the Kaya Symphony system to meet your laboratory needs for throughput, reproducibility, and flexibility. Analytes can be used for downstream applications such as next-generation sequencing and bioinformatics, digital PCR, and real-time qPCR. It's that simple. Now you are ready to do multi-analyte testing on your own. Make the most out of precious samples by isolating multiple analytes. The intuitive workflow uses our sample technologies and automated solutions that are tailor-fit to your laboratory needs. Lastly, the streamlined workflow enables more insight generation for biomarker discovery and validation. Each analyte provides unique insights crucial in providing a complete picture of cancer. These insights are instrumental in revolutionizing how liquid biopsy is used for cancer research.
Can you give us a real-world example where the multi-analyte approach has made a tangible difference in someone's workflow?
Yeah, absolutely. I think the ELIMA-1 study is a good example. The study was done by Dr. Corinna Keub and Professor Sabine Casimir-Bauer from the University of Essen. And there they looked into patients with metastatic breast cancer. They were isolating exactly these analytes I've just mentioned in the workflow, so starting with circulating tumor cells, free circulating DNA and exosomal RNA. And all of these analytes actually help to puzzle the picture together of what's going on those patients.
That indeed provides a very detailed and comprehensive view, right, of the sample. So that about sums up the sample preparation for some blood, but this shows about liquid biopsies in general. So can you share an example of a sample that is less invasive, something that you're developing in this direction?
Yeah, absolutely. I think urine is actually developing into a very interesting analyte.
Right.
So we just launched a PAX-gene liquid biopsy set for urine to isolate free circulating DNA. Urine is especially of interest for all the urogenital diseases like kidney, bladder cancer, prostate cancer, but it also gives insights into tumor entities which are north of the kidney, let me put it this way. And there we see quite a bit of traction and research going on, especially since you can sample urine repetitively. You can get large volumes of urine, and that allows really also to follow up on disease status.
Right. That's some really exciting research going on there. Well, hold on to that thought, Marcus. And this is probably also the right time in the show where we can move on to the next part of the analysis, right? The sample analysis part of the workflow. Thanks so much for being here today with us, Marcus. Would you have some time to stick around for a little longer so we can take some questions at the end from the audience?
Absolutely, my pleasure.
Right. All right. So moving on to the next step of the workflow, I have a new guest with me. Jonathan Scheffer is heading up the NGS research and development team here at QIAGEN. He'll be joining us digitally today. So welcome, Jonathan.
Thanks, Karchi. Glad to be here.
Right. So before you joined, Jonathan, Marcus and I basically spoke about practical SOPs of handling multi-analytes and liquid biopsy. Now we are trying to shift the gears to talking about more the sample analysis part. As we all know, the field is evolving. There's a lot of demand to integrate data from DNA, RNA, epigenetics, and even proteins. So what's the story there?
Sure, absolutely. So biology is inherently complex. You throw in diseases and biology becomes even more complex. It's very important to understand DNA, RNA, epigenetic modifications, and protein. And you can use these to untangle the web of biological complexity. Furthermore, when you look at what's important for next generation sequencing, it's very important to maximize your time usage and also minimize the usage of your samples, because everything is very important when it comes to your biological sample, particularly liquid biopsies.
So speaking of untangling the web, how do you easily manage the different analytes together? I normally think DNA and RNA are analyzed separately because they don't really mix, right?
That's where our QIAseq multimodal DNA RNA library kit with optional hybrid capture is gaining a lot of attention lately. The kit simultaneously prepares DNA and RNA from a single total nucleic acid sample. What's great is it can even be multiplexed on a sequencer with either methyl or protein via NGS libraries to increase the modalities that you're researching.
So that's really one kit that's acting as an enabler. That's already promising, right?
It is. And if you can do NGS library prep, you can do this. You can prepare a single whole genome and whole transcriptome library in one day, and you can either proceed to sequencing or you can perform hybrid capture. It truly takes two traditionally separate workflows and collapses them into one.
So it's really one kit, one workflow that works for both DNA and RNA analysis together.
That's right. And it makes sure that both DNA and RNA libraries are prepared for sequencing. And it takes the lab work and really streamlines it and allows researchers to, instead of focusing on a very long library prep, to instead focus on what will be coming downstream of their library prep.
So for our more visual audience, let's take a look at this.
When you run a sequencing lab or molecular pathology lab using NGS, you could end up with multiple workflows in no time. And multiple workflows mean more of everything: personnel, time, cost, and not to mention the added complexity. That's where we come in. Introducing QIAseq Multimodal DNA RNA Library Kit. It consolidates DNA and RNA library prep into a single workflow. Start with total nucleic acid isolation from any material: blood, cells, tissues, saliva, FFP, or cfDNA. From there, library prep is as fast and easy as it gets. Go from total nucleic acids to sequencing-ready libraries in 6 hours, thanks to the integration of multiple technologies in one convenient kit. Want DNA only or RNA only libraries? No problem. You have the flexibility to prepare DNA and RNA, DNA only, or RNA only libraries from a single sample. What's more, the whole genome and whole transcriptome libraries are compatible with further target enrichment by hybrid capture. So, no matter how complicated your multi-omic application is, you can now get your library prep done using QIAseq Multimodal DNA RNA Library Kit.
So when you combine the QIAseq multimodal approach with the DNA and RNA isolation steps that I spoke to Marcus about a little while ago, that should really be saving a lot of stress in the research workflow.
It does. And what the QIAseq Multimodal DNA RNA Library Kit can do in combination with the hybrid capture panels, it allows you to select what biomarkers you want to work on. On the RNA side, you can look at gene expression, exon skipping, and fusion detection. And on the DNA side, you can look at single nucleotide variants, copy number variations, insertions, and deletions. You can also measure tumor mutation burden, homologous recombination deficiency, and microsatellite instability. And truly, it gives you the option to combine modalities together and utilize QIAGEN's digital insights downstream to focus on the aspects that you want to.
So what would you say, what makes the QIAseq kits and the QIAseq hybrid capture panels so uniquely suited for these kind of workflows? Because I can imagine, working with liquid biopsies is often, confronted with low target nucleic acid concentrations or, for example, fragmented nucleic acids. So how do we go about that?
Great point. And really, very importantly, the QIAseq kit enables you to start with only 10 nanograms of nucleic acid. And in addition, the QIAseq multimodal kit is really modular.
Another M word there.
Yeah, it's really funny that something that's multimodal can also be modular. You can either do whole genome and whole transcriptome, you can do whole genome alone, whole transcriptome alone, but then when you get to the targeted panels, you don't have to look at DNA and RNA together. You can have panels that are specific for what you're looking at. For example, you can have an RNA panel that's only specific for fusions, and you can have a DNA panel that's only specific for, say, single nucleotide variants or copy number variations, and you can really use those independently of each other. And it really lets you just choose exactly what you need for your experimental setup.
So, multi-analyte analysis from start to finish. Can you give us a little synopsis for our audience, Jonathan?
I'd be glad to. So, you first start with 2 PAX-gene cfDNA tubes, and you isolate your nucleic acids for both DNA and RNA. From there, you, in one library prep kit, perform your whole genome and whole transcriptome library prep, and you can perform either then sequencing or hybrid capture, say with our comprehensive genomic profiling panel. After that, you can use our QIAGEN Digital Insights to analyze your data. And truly start to finish, you can use the QIAGEN solutions to profile liquid biopsies.
You make that sound so easy.
In principle, it sounds easy, but in practice, biology is complex. Tumors are very complex. And what we've done is streamline the lab portion of this to allow researchers to not only focus on the results, but focus on their interpretation and focus on what's next. So, in an essence, we've decomplicated one aspect of the workflow, but in the grand scheme of things, biology is still complex and there's still a lot to be answered.
Agreed. And with that, we are really coming to the close of this segment. Would you be able to stick around to answer some of the questions from our audience?
Yes, absolutely.
Well, thank you so much for your engagement and sending in all these exciting questions. We'll be taking a few of those right away. So the first one here, which trends are you seeing on the multimodal testing horizon? Are there any additional analytes being added in multimodal testing workflows? Marcus, do you want to go with that?
Yeah, sure. Let me give the first shot. So I think on top of what we just have discussed, there's also other analytes or methods being used. So microRNA for sure is a topic. Methylation epigenetics, Jonathan has mentioned that already. But on top of this, we also see like nucleosome positioning or fragmentomics is a new field of interest. Genes which are actively transcribed have a different nucleosome structure and therefore also end up in different fragment patterns. So people start looking into these patterns to interpret on what's going on. These are just a few examples.
Right. Okay. Let's take a look at another one. And among the various testing options, which analyte is the most important for the overall picture? I think both of you can answer that. I guess, Jonathan, you should go first maybe, and then we'll switch over to Marcus. Yeah.
Sure. And what we see as a trend is that DNA is more or less the backbone. So you're looking at single nucleotide variants, but other analytes are being layered on over time. Particularly RNA fusions have a place. And as Marcus mentioned, you can add microRNAs, you can add gene expression, you can add a whole host of other analytes, and maybe Marcus would like to add on to that.
Yeah, I think the answer is also not black and white. I feel it really depends on the cancer entity. For example, in the ELIMA study we've just discussed before, there the fraction of circulating tumor cells, the genomic profile we were generating from those, and also the transcription profile of the circulating tumor cells was adding the biggest piece of the puzzle. But I think it really depends on the case. In this study, it was metastatic breast cancer, so there were quite a few cells circulating in the blood, and it can be really different if you look at different cancer entities or other diseases.
Yeah, the world of liquid biopsy is indeed so diverse. I think we have time for one last question. I'm going to take one more. In the long run, do you see liquid biopsies replacing conventional tissue biopsies, or will multimodal liquid biopsy testing always be used as an additional source of information? I guess that's one for you, Jonathan.
Great question. And to play off what Marcus just answered, it's not exactly black or white. So if you have a tissue biopsy, you can always use that tissue biopsy, and liquid biopsies won't truly replace that. But for various types of cancers or various types of diseases, when you have access to liquid biopsy, whether that's, say, blood or saliva or urine, that you're able to utilize that potentially as a surrogate to prevent having to do biopsies in all contexts. And again, it comes down to your disease and cancer of interest, but in the end, it still truly depends. And that really, harkens back to the idea that biology is very complex and there's not a one-size-fits-all answer.
Yeah. So maybe to add on this, it also depends on the organ or on the disease. For example, brain cancer, you usually don't take biopsies. Another example is, for example, after surgery, when the tumor is gone, and you do minimal residual disease follow-up. If the treatment is successful, then, for example, liquid biopsy is the only option you have on top of visual investigations. So there are fields which are really where liquid biopsy will be dominant, and others where still physical biopsy will need to be taken.
Thank you. With this, we really come to the end of the show. Thank you so much for being here today, both of you. I learned a lot, and I hope our audience did it too. Join us next time for Q-rious, Simplify, Accelerate, and Detect. We are going to talk about digital PCR in oncology and translational research. Thanks for watching.